This is Christine McGowan, and I'm going to be doing the DNA extraction lab. So to begin with, DNA, or deoxyribonucleic acid, is a complex molecule that contains the genetic instructions in the development and function of all living organisms. It's a type of nucleic acid that consists of long chains of nucleotides that each contain a sugar molecule, a phosphate group, and a base, nitrogenous base. The nitrogenous bases are adenine, thymine, cytosine, and guanine, and the order of these bases determines the genetic code that gets passed down from generation to generation. DNA plays a crucial role in living organisms as it contains the instructions for the synthesis of proteins, which all cells need. The process by which DNA is used to synthesize proteins is known as gene expression. It's also important in the replication and transmission of genetic information from one generation to the next. Okay, so I'm going to begin by taking the split peas that have been soaking in distilled water. I'm going to add them to the mortar and pestle. And then I'm going to add some salt to some distilled water. And then we're going to add that to the mortar and pestle to try to break down the cell membranes outside so that we are able to extract the nucleus, or extract the DNA from the nucleus of the cell. Okay, so I'm going to stir the salt water so that it's completely dissolved. And then we're going to add a little bit till about halfway, and then we're going to break down. Okay, so we've broken down the split peas pretty well, and we're going to pour the contents into another cup, but we're going to strain it with cheesecloth first. Okay, so to this now solution, this is the extracted solution from the split peas. We're going to add some detergent here. And the reason we do this is because the detergent is going to also aid in helping down to break down the nuclear membrane. Detergent is amphipathic, meaning it can interact with hydrophobic and hydrophilic molecules. So to do that, it's going to be able to disrupt those membranes to make the DNA extraction easier. I'm going to add a little bit more. Perfect. And then we're going to put this in the fridge and let it cool for 10 minutes. Okay, so the solution has been sitting in the refrigerator for 10 minutes. We're going to pour this solution into a test tube until it's about 1/3 of the way full. Okay. Now we're going to add the papain powder, or the meat tenderizer, and this is going to help to break down some of the proteins that are surrounding the DNA. And then we're going to stir it very carefully. And now we're going to add some of the ice cold isopropyl alcohol. This is going to help to precipitate the DNA out of the solution. And if we look, we can see the white stringy DNA filaments that are in here that would indicate that we have successfully extracted the DNA.